Hello everyone. I am Sudhanva Kamath. I am a PhD student at the Indian Institute of Science. I will talk to you all about my work on telomere-to-telomere assembly by preserving contained reads. Genome assembly involves identifying information contained in the suffix-prefix overlaps between pairs of reads to assemble the unknown chromosomal sequences in the sample's genome. In the era of long read sequencing, three types of sequencing data stand out for their read lengths and per base accuracy. Pacific Biosciences high fidelity reads, Oxford Nanopore Technologies simplex reads, and ONT duplex reads. Notably, PacBio hi-fi reads
Hi,
have a high.
sorry. I think we are not seeing your presentation, the slides moving forward.
Oh,
We are still seeing your first slide.
I see. Let me just check. I'll just stop and share once again.
Okay, thank you. Okay, so apparently people online see your presentation moving forward, but we are not seeing it here, so we are gonna
Okay.
check.
Um,
Can you try again?
yeah.
Can you refresh and try again?
So I'm
Yeah.
just changing slides. Is it visible now?
Yeah, can you maybe move one ahead to see whether it changes? And yeah, now it's working. Thank you so much.
Okay. Um, yeah. So notably, PacBio hi-fi reads have a higher per base accuracy at 99.8% and a smaller range of read lengths. So genome assembly is performed by first building a graph-based representation of the overlap information contained in the sequencing data, and three data structures are commonly used: the De Bruijn graph, overlap graphs, and string graphs. So I will focus on overlap graphs and string graphs today. In the overlap graphs, the vertex set is the set of all reads, and the edges are added between a pair of vertices if there is a suffix prefix overlap of sufficient length, let's say k. A string graph is a sparsified subgraph of the overlap graph, and two heuristics are used to sparsify the overlap graph. All transitive edges are removed, and this heuristic has actually been shown to be safe by Jain. And I will focus on the other heuristic, which is the deletion of all contained reads, which leads to the deletion of all vertices corresponding to those reads and edges associated with them. This operation is not safe and has been shown to lead to loss of information relevant to accurate and complete genome assembly. So why use string graphs at all? So string graphs actually have a much simpler topology compared to overlap graphs. So this is an overlap graph of the chromosome 20 of some reads simulated from chromosome 20 of the CHM13 reference genome. And af- after application of the two heuristics, we end up with a much simpler string graph. So how does contained read deletion actually impact accurate genome assembly? Let's consider a diploid genome and read sampled from a location near a heterozygous allele. You can see in this toy example, the contained read R6 after deletion leaves this region unsupported on the second haplotype. And the string graph has only one valid path in this region. This leads to only one copy of the heterozygous allele being assembled in both contigs in this region. This is an incorrect assembly. So we aim to tackle the problem of contained read deletion in this work by answering two key questions. How frequently do we see such gaps? And how do we make long read assemblers resilient to contained read deletion? To answer the first question, let's first set out our objective. We want to identify the we want to analyze the impact of the depth of sequencing coverage and choice of sequencing technology, parameters which we can control, on the likelihood of observing a gap. The challenge lies in the fact that many different factors can influence the frequency of observing a gap, some of which are just where we sample the reads from, what are the read lengths, what is the heterozygosity rate in the genome, and so on. So let's start with a simple model. We'll assume a diploid circular genome uh, to avoid edge effects, which contains a single pair of chromosomes of length G. And we'll assume that there is a single SNP locus at position 1 and that there are no repetitive sequences in the genome. A read is simply a substring of one of the haplotype sequences, and two reads are said to be equivalent if they spell the same genomic string. So each read is characterized by its haplotype of origin, length, and starting position. We assume that the reads are sampled uniformly at random, and the read sequencing output is simply a pair of multisets S1, S2, Si being a multiset of reads from haplotype I. The user will provide the depth of sequencing coverage and read length distribution, which corresponds to the choice of sequencing technology as input. The frequency of observing gap is simply the number of sequencing outputs which contain a gap to the number of all divided by the number of all sequencing outputs. So how do we define an assembly gap due to contained read deletion? When we have a sequencing output, which when we have a particular sequencing output, we will only restrict our attention to those outputs where both the heterozygous alleles have been sequenced on, when both the heterozygous alleles have been sequenced. So we will actually have the luxury of considering two parameters, x1 and x2, xi being the stop position of that read which contains the heterozygous allele on haplotype I and extends furthest towards the position lambda, lambda being the length of the longest read in the sequencing output. Since x1 and x2 are well defined for any sequencing output, we can partition all sequencing outputs into classes labeled x1, x2. And then we will define a sequencing output belonging to class x1, x2. When does it contain an assembly gap due to contained read deletion? Specifically, we will look at the interval minimum x1, x2, minimum x1, x2 plus 1. If this interval is supported on both haplotypes originally, and after contained read deletion, it is not supported on at least one haplotype, then we will say that there is an assembly gap due to contained read deletion. So how do we count all the outputs that actually contain a assembly gap? So we will do that by characterizing all sequencing outputs in class x1, x2 that contain an assembly gap due to contained read deletion. And we achieve that with this lemma. Three conditions are I have identified three conditions which will guarantee that a sequencing output contains an assembly gap. So first condition guarantees that the support for the relevant interval can potentially be lost. The second condition guarantees that a contained read which originally supported the relevant interval, minimum x1, x2, minimum x1, x2 plus 1, is being deleted. And the third condition guarantees that no read supports the relevant interval after contained read deletion. So these three conditions will actually guarantee that there is a assembly gap. But how do we still need a proof of necessity, and I will just sketch a brief proof. To argue necessity, we'll just violate each of the conditions and argue that an assembly gap is not possible. If x1 is equal to x2, then either x1, x1 plus 1 is supported before contained read deletion and after contained read deletion, or it wasn't supported before contained read deletion and it remains unsupported after contained read deletion. For condition 2, if x1 is greater than x2, then if there is no contained read in which supports both x2 and x2 plus 1, then that interval will remain unsupported both before and after the deletion of contained reads, and then there is no assembly gap. Lastly, if there is no read which starts before x2 and extends past x1, like read R9 here, uh, the existence of read R9 will guarantee that x2, x2 plus 1 will be supported both before and after contained read deletion on both haplotypes, meaning that there won't be an assembly gap due to contained read deletion. So having characterized all sequencing outputs in class x1, x2 containing assembly gaps, we will count them using generating functions represent for every read for all valid choices of x1, x2. The fraction of outputs containing a gap is the frequency of observing a gap due to contained read deletion. In our experiment, we varied the sequencing depth from 20x to 100x in total. So that would be 10x on each haplotype to 50x on each haplotype. We expect gaps to become less frequent when the depth of sequencing coverage is high. And this is because the likelihood of every read supporting a particular genomic interval being contained in a becoming a contained read decreases as coverage increases. We also expect that gaps will occur more frequently if the range of read lengths is large. And that is actually what we observe with ONT reads having order of magnitude higher likelihood of observing a gap. So we've answered our first question, how frequently do gaps occur? So how do we solve our assembly problem? So if all reads were of the same length, then this problem would not occur. So we could just fragment all reads into equal sized pieces. But this would ignore the possibility that there are repetitive regions in the genome, which long reads are very good at spanning, which assemblers uh, find it easier to assemble regions containing repeats if there exist reads which span the entire repetitive region. So we planned a repeat aware fragmentation procedure where long reads which contain repetitive regions, we will leave them unfragmented as much as possible. So how do we identify these reads which contain these repetitive regions? The idea is to use the alignment information contained in all versus all overlaps to identify how many reads align to a particular location on the read. If that number is roughly equal to the estimated coverage, then we fragment that read as though it was part of a non-repetitive region. If we identify a region which is which is part of a repetitive region, which we identify by recognizing a significantly higher number of reads aligning to that particular location, we leave that region unfragmented and only fragment the non-repetitive regions. And the impact on the read length distribution is immediate. This is a ONT duplex dataset of real reads, and the range of read lengths drops from 170,000 to 60,000. We propose the following assembly pipeline where we use our tool Raft. We created our pipeline along with HiFi ASM, a string graph based assembler, and we identified that the number of assembly gaps due to contained deletion across 5 simulated datasets drops to single digit values as compared to other methods. We have also assembled real datasets, 4 of them, one PacBio HiFi dataset, one ONT duplex dataset, a third dataset comprising reads from both of the first 2 datasets, and lastly a high accuracy ultra long ONT simplex dataset. In any dataset which contains ONT reads, we observe that the Raft HiFi ASM method doubles the assembly contiguity and also generates telomere-to-telomere contigs at with no changes to the switch error rate or the gene completeness values. Raft HiFi ASM, the increase in assembly contiguity is demonstrated here in ideograms of contigs generated by HiFi ASM and Raft HiFi ASM. And I would like to say that Raft HiFi ASM has been used by Oxford Nanopore Technologies to generate a highly contiguous assembly of HG002 along with additional complementary parental data. I would like to conclude my presentation by saying that contained deletion is still not a solved problem and remains a major bottleneck for string graph based assemblers. But we have demonstrated that assembly gaps due to contained deletion occur more frequently in datasets with a large range of read lengths. Using Raft reduces the number of assembly gaps, and there is also a twofold increase in assembly contiguity when using Raft HiFi ASM. We have created 2 open source tools, CGProb for our theoretical model for assembly gaps and Raft for fragmenting reads. Both of them are available on GitHub. Our preprint for this work is available on bioRxiv and is currently under revision at Genome Research. Thank you.
Thank you so much for your presentation. Do we have, maybe you can, you can.
So I was wondering, are you able to use your method to call variation within, or could you sort of change your method to detect variation and like more accurate, increase the accuracy of of the detection of genetic variants?
So our method is intended for de novo genome assembly. I assume you're talking about Raft HiFi ASM.
Yeah, if you could sort of take some of the main ideas from it and like look at sort of using it for the detection of variants.
So I think current methods for I can't comment on the other work, but our work is intended for a de novo assembly. So the idea is that if we get more accurate assemblies, they can be then used for better variant calling. And we have actually observed that there is a slight increase in overall variant calling for SNPs and indels when using the Raft HiFi ASM method. So you can see that
Oh,
the
okay.
F1 scores do increase. However, these variants were called using dip call,
Oh, okay.
not using the reads themselves. We assembled the reads first and then called variants.
So in the improvement of the gaps within the variants, did you restrict interest to the gaps of specific sizes?
Um, No. So any and all contained reads which might potentially create a gap, uh, we aim to solve that problem. However, what we do is we do restrict our attention to uh, reads. The reads that we fragment them to 20,000 base pairs. This is a configurable parameter, but we found that for HE002 and other human genomes, we expect that we will get good results with 20,000. uh, we believe that this is because of the different types of repeats that exist in the human genome. And what we want to do is ensure that repeats of specific lengths are still resolved. So that is also a parameter which can be tweaked in Raft.
Okay.